Good evening, everyone. I Manoj Kumar Dubey welcome all in third lecture on Atmanirbhar Bharat lecture series 2025. Today's lecture is based on Nobel Prize in Chemistry 2024. I feel privileged to welcome Honorable President Sir of IHS, Dr. Suman Kumar Mishra, Manager Ma'am, Mrs. Sandhya Mishra, Academic Advisor, Mrs. Jayanti Samant Ma'am, Principal Sir, Mr. Santosh Kumar Srivastav, Vice Principal Sir, Mr. Ganesh Shankar Chaturvedi Sir, and Head Mistress, Mrs. Anupma Srivastav. Dear teachers and my beloved students, this evening is going to be full of learning in irrespective of your stream you belong to. The speaker of today's lecture are Ms. Mahashweta, Faculty of IHS PGT Chemistry, and Mrs. Anupam Srivastava, Headmistress, International Hindu School. All the participants, please take care. If few things before we start the meeting of lecture series. First, everyone should keep themselves mute during the lecture series. Second point, if possible, all of you are requested to keep your video on during the lecture series. And third one is very important. During the question and answer session, question can be asked through chat box only. Now I would like to request Honorable President Sir to speak few words and consent us to start this lecture series.
Thank you. I'm very happy to be part of this lecture series. We have been doing the Nobel Prize lecture series for several years now, and this is our very, very laudable series. We have had speakers from around the globe in the last several years, people from England, Australia, from America, and they have participated in this series. So this lecture series is important because Atmanirbhar Bharat, self-sufficient India, cannot be without a rapid progress in the science. We need to have more students take basic science research. We need to solve the problems. Otherwise, if we don't solve our own problems, we'll always be second rate. We'll be followers of somebody else, of the West. If we want to lead our own growth, we have to find the solutions of our own problems. These Nobel Prize winners, they have worked all their life to win a Nobel Prize, and they found something very unique, that it helped the humanity solve the problems of the whole humankind. Would you believe that the money for this Nobel Prize, it comes from the bombs which we explode. The explosion of the bomb, he discovered that, Mr. Nobel, and gave the rights to everybody. He did not keep the rights himself. So that money is piled up, and that's the Academy in Sweden. They give all these prizes. So without further ado, I'd invite Mahashweta Mukherjee to give us the lecture. Thank you.
So, good evening, everyone. This is Mahashweta Mukherjee, PGT Chemistry, International Hindu School. On behalf of Chemistry Department, International Hindu School, I would be delivering lecture on Nobel Prize in Chemistry 2024. The theme of this Nobel Prize lecture series is Atmanirbhar Bharat. And the topic of this lecture series, the Nobel Prize in Chemistry 2024 was awarded for computational protein design and protein structure prediction. Okay, so before starting this, let me tell something in brief about proteins. So actually, all this protein is very important in our real life. Our hair, our nail, everything is made up of protein. Normally, all of must have had fever someday or other. So what happens? The doctors advise us that you should take paracetamol. So this is because when the temperature rises, temperature of our body rises, all our body is made up of proteins somewhere or the other, be it hemoglobin, our hair, or our enzymes. So when the temperature rises, the active sites of the enzymes gets denatured. So if the enzymes won't function, obviously and if the fever will rise up to a certain limit, it might cause death of the individual. So that's why this protein is very important. So let us see who got the Nobel Prize in Chemistry 2024. Okay. So here are the Nobel laureates. So Sir David Becker, he is from University of Washington. He got Nobel Prize for computational protein design. Using biochemical techniques, he has designed proteins. And the proteins designed by him are very important. Some of his proteins have worked as nanofibers in medicine. Also, some of his protein has uh, helped in treating cancer, while Sir Demis Hassabis and Sir John M. Jumper have worked on protein structure prediction. So actually, before understanding what they did, like how they predicted protein structure and what is the problems which were being faced in predicting the protein structure, we will discuss structure of protein. So here is a brief introduction about them. So as I already said that Sir David Becker, he's from University of Washington and he's an American biochemist and computational biologist. So he has worked on computational chemistry and with the help of that, he has synthesized many proteins. Okay. Sir Demis Hassabis and John Jumper, they are co-founders of Google DeepMind and they have used AI model AlphaFold 2, about which I'll be discussing in the latter part of this lecture. So he has designed, as mentioned in the slide, he has designed smart protein drug, which has helped in cancer treatment. So in biochemistry, there is a saying that if you want to know the function of protein, first of all, you must understand the structure of protein. So over here, you can see primary structure, amino acid sequence. So these bead-like model, you can see the colored models. They are actually colored beads. They are actually amino acids. Okay. So they are the amino acids which are joined together to form the primary structure. Now, coming to the secondary structure, which consists of alpha helix and beta sheet. So here I would like to explain the alpha helix and beta sheet with this. So see, you might imagine this as amino acid ka sequence. Suppose amino acids are here like beads. Okay. So if I try to curve this is the alpha helix. So here I am representing the secondary structure. So you can see this is the alpha helix over here. Okay. When it is arranged in this way. Further, the part of the secondary structure also consists of beta pleated sheath. So I have used this paper to explain this. It looks like this. This is beta pleated sheath. So this is the secondary structure of protein. And now when the beta alpha helix and beta sheath, they combine together, it forms the tertiary structure. And when many such structures combine together, they form the quaternary structure. Now, what was the problem which was being faced earlier? So, we actually use, now also, we actually use NMR spectroscopy, X-ray crystallography, and cryo-EM. So these techniques actually are very costly because not all universities have access to these instruments. Plus, if you want to get results by them, it takes years of time. But we can find the structure of amino acid using that. But now you can see, I have explained that the protein structure is such three-dimensional and it is very complex. So if you want to explain it using or understand using NMR or cryo-EM, so it will waste a lot of time. So actually this problem has been solved by this AlphaFold by the scientists, that is Sir John Jumper and Sir Demis Hassabis. So let us move forward. So see, this is some description about the protein. So proteins are chains of amino acid. As I already showed you using this realia, that they are actually made up of amino acids which are joined together. So these are building blocks of our body, our hair, our nail, even the hemoglobin, which is the carrier of oxygen, is actually protein. The protein has three-dimensional structure and different proteins have different amino acid sequence. Apart from that, the proteins are enzymes which are essential for our normal functioning of our life. Okay, like proteases helps in breaking proteins. Apart from that, the proteins provide they are structural components of our tissue. Like of muscles, actin and myosins are the structural components. Hormones are also protein, like follicle stimulating hormone or insulin. Insulin is a hormone about which we all might have heard. It lowers the glucose level. Apart from that, the antibodies, which are responsible for defense mechanism in our body, are also proteins. So by this, we can understand that protein is so much important. Now, in this slide, I have represented that how proteins are important. So you can see over here, digestive enzymes help facilitate chemical reaction. Digestive enzymes are there like proteases, which helps in breaking proteins into smaller components. All the enzymes collaboratively work together and they help in effective digestion. And proteins, actin and myosin, they are the muscle fibers. They help in contraction, movement, all that we walk or we are able to pick any object. It is due to this actin and myosin. Apart from that, collagen, elastin, they provide support to our body. They are also proteins. Antibodies are proteins like IgA, immunoglobulin A, immunoglobulin G. They also are proteins and they help in defense of our body. Hormones are there such as insulin, follicle stimulating hormones. They are also proteins. Apart from that, obviously DNA is made up of protein. DNA has histones in it, which provide its structure. That is also a protein. Hemoglobin is also a protein. So the oxygen we breathe in is carried in our body through the protein pigment called hemoglobin. So that's the importance of protein, because even if we are able to inhale oxygen, suppose it doesn't reach to all the organs or our heart, then obviously we might not be able to survive. Thus, the protein is very important. And thus we can understand that why synthesis of protein or its structural determination is of so much importance. And that's why the Nobel Prize awarded to these laureates is of utmost importance. So, Sir David Becker has worked on discovering proteins which were not earlier found in nature. As I have already discussed, that he has found a protein which targets only the cancerous cells. we already know that the patient dies due to the treatment of cancer, because while getting treated, what happens that the normal cells are also getting killed. And Sir David Baker has worked on that. This research work actually started in 2003. When he first made a protein, he tried to see whether the structure is coming correct or not. So he had firstly synthesized a protein, and after he synthesized the protein using the amino acid sequence, he did it biochemically. After that, he found the structure using AI-driven software, that is Rosetta. How, like, he has also discovered certain proteins which can inhibit the functioning of different virus by sticking on the spikes of the viruses. Also, some proteins can catalyze the reactions. He has also discovered certain proteins. And he has, obviously, as protein is of so much important, so his founding is it is a boon for us. Okay. And he has also synthesized a vaccine of COVID-19, which was of great help. He, along with his colleagues of University of Washington, has worked on this. Now, these are different proteins which were synthesized by Sir David Baker. So he has made artificial proteins. He has studied protein-protein interaction, like how the two proteins are interacting. You can say like antibody, antigen. He has studied some effects of genetic variations, genetic variants. Actually, the protein mutation also causes some genetic diseases. It's about that. He has discovered many drugs, like the drug for cancer treatment. Now, in this slide, we are able to see the proteins which were designed by Sir David Baker. So in 2011, he had designed an antiviral protein against influenza. So that time, influenza was a very prominent disease, like people were all over affected. So he had designed an antiviral protein. Also, he has designed a protein nanoparticle, like he has used nanoparticle in solving many problems, like in drug action. So those nanoparticles are very conducting, so they could reach the target and they were acting very efficiently because of their small size. Also, during the COVID, he has made many inhibitors which will inhibit the actions of protein by sticking on the spikes of the virus. Also, yeah, he has designed nanofibers. Nanofibers are small tubes which will, suppose it will act on the target areas, target tissues, and thus it will enhance the process of healing. Using that, we can also give drugs using these thin nanofibers at the target areas where the wound is there. So after talking about Sir David Baker, let us talk something about Sir Demis Hassabis and Sir John Jumper. As I already showed you or tried to explain, that the protein structure is three-dimensional and it is very complex. So if we want to find the structure of amino acid, it only takes years. Now, if we are going to use NMR spectroscopy, cryo-EM, or X-ray crystallography, so the protein structure prediction will be very chaotic. It will take years and years of time. So to solve it, actually, earlier also they were working on this. That's what AlphaFold 1. Earlier also they were working, but the accuracy was 40%. But this time they have discovered an AI-driven project, AlphaFold 2. So what will happen that it's not like we will not give it some information. We will provide it the amino acid, like which amino acid is to be used. we will feed the data, and the AlphaFold 2 will use neural networking. Using the neural networking, it will use the amino acids. So it is to be understood that the structure of amino acids would be determined by NMR or IR, NMR or X-ray crystallography. And after knowing the structure of amino acids, we will just feed the data in the AlphaFold 2. It will use the neural networking, and using that, it will provide us the structure of the probable structure of protein. See, it is to be noted that even that structure would be only 90% accurate. But the thing is that all this is saving such a lot of time. Otherwise, it would have taken years and years to predict a single structure of a simple protein. So now it can be done within days. Plus, the accuracy is also 90%. Apart from that, if you want to determine a structure using these techniques, you need instruments which are very costly. So this is not cost effective, and all this is not accessible to everyone. But the AlphaFold 2, it can be used for determining the structure of proteins easily. So apart from that, they have also worked on synthesizing an enzyme which decompose plastic. So plastic is a hazard for all of us, for the environment. So he has worked on it. They have collaborated, like they have worked on it together, and they have created an enzyme like cutinase, which will help to decompose plastic. And through this, we could understand. Now we are able to relate why the structure prediction of protein is so important, and what a boon is the AlphaFold 2 for us, especially for the scientists who are working on drugs or who are working for providing vaccines. Because we never know, any like disease like corona can come again. So we should be prepared, and this will be very helpful for us. So you can see these were the techniques which were earlier being used. Like it's not like it is not now used, it is also used, but it just takes a lot of time. So the AlphaFold 2 has helped in replacing this, and it has helped to predict the structure of protein efficiently. So in this slide, I have just shown how this neural networking functions, like how AlphaFold 2 works. So actually we have to input the data, like which amino acids are there. And using neural networking, it will try to predict. So these beads over here is actually the beads over here, you can see, are actually the amino acids. So these dark ones, these just predict that these yellow on the X, you can see that if this is X-axis and this is Y-axis, these dark blocks, this just predict that these amino acids are close. So using the study, they have just tried to predict the structure of proteins, which is 90% accurate, but they are also working on it to make it more efficient, and so that we can determine it more easily. Thank you, everyone.
Thank you, Mahashweta ma'am. Now I would like to invite Mrs. Anupam Srivastava ma'am to speak few words on the topic.
Thank you, Manoj sir, for giving me opportunity. Once again, with due respect to Honorable President sir, Principal sir, Vice Principal sir, Manager ma'am, Advisor ma'am, dear teachers and my lovely students, I am here to talk on the implications of Nobel Prize in Chemistry 2024. As you have already seen that the Nobel Prize in Chemistry 2024 has been divided between three people, David Barker, Demis Hassabis, and John Jumper. The contribution of David Barker is that he talked about the computational protein design. The contribution of Demis Hassabis and John Jumper is that they have given the AI solutions. If we talk about the implications, when we say implications, children, implications means uses. What are the benefits? What are the importance of these prizes or these contribution to India and other countries? So the three main importance are there in the field of agriculture, in the field of medicine, and in the field of ecology or the ecosystem. First of all, I will talk about the implications in the field of medicine. Accelerated drug discovery and development, reduce healthcare costs, advancement in neglected disease and research, growth in biotechnology and nanomaterials, in agriculture and other medicines, boost to AI research system, enhance academic and industrial collaboration. If we talk about in the field of medicine, then diseases like cancer, when they were treated, even the potent cell of the organism used to die due to chemotherapy. But now we can design the targeted medicines which will attack only the carcinogenic cells and the pathogens, so the healthy cells of the organism will not die, and then organism can stay or the person can stay or the patient can live longer. The second thing, in the discovery of these medicines, lot of economical pressure was on the countries. But after this research, now the healthcare cost will reduce. Then there were some more diseases which were totally neglected. The medicine of those diseases were not discovered. Now those medicines can also be developed. In the field of growth and in the field of biotechnology also the work can be done. The nanomaterials can be prepared which help in surgery, etc., and thus the cost can come down and patients can be relieved. In the AI can help to know the proteins and their functions which can work direct. So this will be a great boon in the field of medicine. If we talk about in the field of agriculture, then strong variety of seeds can be developed after knowing the protein structure. We can have crop which have higher resilience. When we say higher resilience, it means they can bear the dot, the drought, and the other pathogens. Thus, we can have large yield. We can have immense growth of the crops. Then it is a boon for the ecosystem. How it is boon for the ecosystem? The whole ecosystem can be benefited. How? Because one of the remarkable discovery they are telling can be done by designing those drugs or those chemicals or those proteins which can directly degrade the plastics. We know plastics are non-biodegradable. Plastics are not degraded by the microorganisms already present. And I have told you before also that plastics, wherever they go, if we dump in the land becomes sterile. So this will be a biggest boon for the ecosystem. The whole ecosystem can be cleared and it will help in green atmosphere, green world, green earth. Then in the field of green chemistry also, many of the chemical processes can now be more with the help of biotechnology, it can be eco-friendly and this will help a lot. So thus we see that this Nobel Prize or this research or this development is a boon for the mankind. India can change the traditional research and development programs in all the sectors and work can work on global landscape with AI solutions. The ability to create proteins that are loaded with new function can lead to rapid growth and development of vaccines and chemical industry. Thus, we can see that this research, this development is really a boon for the mankind. Thank you.
Sitting there, the young girls, they are malnourished and they go straight from youth to old age. There is no adult life for them because they age very fast because of lack of protein. That's one point to make. It's very important to take adequate protein because protein repairs the body. The carbohydrates give you energy, and the fat gives you the storage of the energy so that you can use it when you're not eating. So this is a very simple classification of protein, fat, and carbohydrate. The other important thing for you all, it's most of you now are in 10th, 15 years and higher. But before puberty, there is growth and adequate protein. 1.5 gram per kilogram protein is needed for children during growth because protein not only repairs the wear and tear of the body, but it adds growth to you. Indians are short, many of them, who did not have adequate nourishment during childhood. And I don't want to elaborate on this, but nutrition is very important. Protein is very important to take enough protein and calories during growth. I just want to give you a target for research. You can start thinking about it now. One big problem coming all over the world is dementia. As we get older, our memory goes down. And you know why it is like that? Because when we grow, the number of cells which are produced in the brain, they stop multiplying at age 18. There is no new cell formed by the brain or the nervous system after that. So from then on, it's up to you to protect your cells so that they don't get damaged. But you have to discover. You have to discover a process how the cells can multiply, nerve cells can multiply, even after 18 months. Now, in the previous lecture, I had mentioned that if you are looking for topics for research, study Mahabharata. And I'll tell you a reference to this protein right now. I have read in Mahabharata that people used to live 10,000 years in Satyug, and they used to be 10 foot tall. Why have we shrunk? Our age has come down to 100 years. If we live 100, we have achieved a big landmark. So those are those points. The last point I want to make is this targeted treatment, what we are talking about, that we give an injection and it targets only the culprit cell, the culprit cancer. So you understand antigen antibody. I mean, that is taught in, I think it's taught in 11th and 12th, antigen and antibody immunity. So here we are producing a protein which is attaching to the surface of the recipient. just like antibody-antigen reaction. So in the beginning, there was a problem because I have treated patients with such medicines that you are giving treatment for one thing and something else comes along. This was not realized earlier, but now it's getting narrowed down. I don't want to take up all your time, but this question, I mean, we have lots of time for questions. We should do in a methodical way that the chat box is used. I just want to tell all the participants that if you think of some question, you can email. You can email to Mahashweta ji or your other chemistry department teachers, and they will try to get back to you. This lecture series will be uploaded on the YouTube in a few days, and you'll be able to review the material even there. I will stop now.
This is time to start the question and answer from the participants side. If any questions you have, then you can write in the chat box. I think there is no question in the chat box.
So if there is no question, I'll make a few more comments because there is still time. The Nobel Prize in Chemistry, I want to give you an overview of chemistry. The whole creation is chemistry. Can you imagine this? The whole creation is nothing but chemistry going on. There is a reaction, chemical reaction going on everywhere, physics and chemistry. These two are running this creation. Now, I shouldn't say that. Brahma is is running the creation, but atomic explosions are taking place on the sun, and it's giving us the heat and the light. So this is something chemistry. If you take interest in chemistry, you'll enjoy it because you will be able to find out more things about what's going on. I mean, just imagine, let your imagination go and say, okay, how come the plants grow? If you put a plant near a window, it bends. It goes over to the window. It does not go straight. I mean, simple answer is the sunlight is there, but why is it happening? What is happening to the cells which allow the stem to bend? And the other side is growing more, and the concave side is growing less. I mean, this is a very, how do you cut? There are carnivorous plants, Venus flytrap, and they trap the insects. They are carnivorous. They eat the insects. So how do they know? Okay, now we have some food here inside our basin, and they close the trap on the top. How does it happen? There's so many mysteries. The whole universe is full of, it's amazing. It's a miracle. The whole world, everyday life is a miracle. We go to sleep and we wake up. How does it happen? How does the time clock work in the brain? That if you think that 4 o'clock, I want to get up, I get up 4 o'clock every morning. So before the alarm goes on, I wake up at 4. There is no clock being wound in the brain. How does that clock work? There are so many things. For example, there is a time to sleep. There is a time to wake up. So why do you start feeling sleepy at about 9, 10 o'clock? That's the natural circadian rhythm I'm describing. There are some hormones which are reduced. But what is the signal? What's the clock? How does that work? The cortisol is reduced, growth hormone is reduced, adrenaline is reduced, and then Brahm Muhurt at 4 o'clock, some hormones start. The cortisol goes up, adrenaline goes up, growth hormone goes up, and wakes you up. Time to get up, open your eyes. I mean, there are so many mysteries. So instead of wasting your time on the cell phone, this is the greatest retardant against your growth and study. Don't use the cell phone. Study hard and exercise, proper nourishment, and have a program. Think of yourself like you have been given this time slot. Your parents are helping you to get trained for life. You are a trainee for life. You are being trained to be a citizen. You are being trained to be a mother. You are being trained to be a father. You are being trained to be a citizen. So use this time very, very wisely, because you can spend $1 million and you cannot buy even 1 second. You cannot go back in time. Time only goes forward. So with all those simple advices, I'll stop for the next speaker.
Thank you, sir. Now I would like to invite Principal Sir for vote of thanks.
Okay. Thank you, Mr. Manoj. First, let me make you correct. I am Santosh Kumar Tiwari. I am not Santosh Kumar Srivastav. Okay. Okay. On lighter note, I am said it because you pronounced my name. That was not Tiwari. Okay. Good evening, everyone. It has been such an honor to be the part of this wonderful evening event, Atmanirbhar Bharat lecture series. We all are very thankful to our respected President Sir for encouraging we teachers to organize this wonderful series for we teachers and our students. We'd also like to offer my thanks to my PGTs, Mr. Manoj Dubey and Masweta Mukherjee ma'am. And Masweta Mukherjee ma'am, your lecture was really very fantastic and we all are very impressed. Okay.
Thank you, sir.
And again, I would like to offer my heartfelt thanks to my headmistress ma'am. She also elaborated everything very nicely. We, the people, those who are not from science background, we also got her that lecture that what is the need of this particular invention, this particular finding of the scientists. Okay. Lastly, I would also like to offer my heartfelt thanks to everyone, those who attended this very wonderful lecture, and they also gave us courage to our teachers that they should also come forward with such type of wonderful session. Thank you, everyone.